Okay, it is my pleasure to open the third session of the afternoon. This session is focused on spinal cord injury as a whole body system and the potential benefits of neuromodulation in that context. And in that capacity, I'm very happy to introduce Dr. Klaus Linnman. He is assistant professor at Harvard Medical School, and he directs the Spaulding Neuroimaging Lab and is a fantastic collaborator and scientist in this space. And so, Dr. Linnman, please.
Yep, perfect.
Thank you.
Thanks. I see a few people coming in still, so we'll give them a few minutes. Yeah, okay. Perfect. So thank you very much, Leslie, for inviting me. This has been very informative. I don't work with spinal cord stimulation or neuromodulation yet. I hope to start some projects. So my background is in neuroimaging, and I've been working primarily in chronic pain and psychiatric conditions. And then starting at Mass General Hospital, I moved over to McLean Hospital for a while, then Boston Children's. But the last 6 years now have been at Spaulding, where I've really gotten into using neuroimaging methods to kind of track pathology and see if we can use those as treatment, as metrics of treatment outcomes. All right, so I wanted, inspired this morning, I wanted to start with a little bit of history of medical devices. And this is another medical device that we all are aware of. This is one of the first demonstrations of the Permobile. This is, I believe, 1969. It was a pretty competent chair back then, could do curves. And this is Per Udén, which is the founder of Permobile. And one of the young engineers you'll see driving the chair is actually my father, who developed the steering mechanisms or the electronics to control these wheelchairs. And I think, Permobile has made, from the 60s, made such a big impact in, establishing both, giving mobility back, but also empowering patients very well. So let's not forget about the kind of basic mechanical or electronic. That's my dad right there in the 60s. So I had the distinct pleasure of piloting a lot of these chairs as a kid. And that's the Permobile Minior, which was the first pediatric wheelchair that came out in around 1985, I believe. So now, 40 years later, I am working with neuroimaging of spinal cord injuries, so kind of closing that loop. And as I said, using neuroimaging to define pathology and also seeing if we can track some of the treatment interventions using maybe more objective markers than just, pain ratings or patient reports. So first question is, how complete are these spinal cord injuries? In some cases, you have a pretty dramatic traumatic injury, so you know that there's very little chance of any residual axons that pass, that can transmit information from the brain to the body or from the body back to the brain. Whereas other cases, here you just see a slight shadow on the cord, but this patient is still an ASIA-A, complete injury, clinically. So the question is, are there residual axons that can pass through the cord, through this glial scar, and is there a way to identify these regions or these potential residual connections? One very simple way of doing that is just the Jindrasic maneuver, where you basically clench your teeth and pull your hands apart so you overstimulate the motor outflow from the brainstem. And that can lead to an enhanced patellar reflex, both in able-bodied subjects, but also in some patients with complete spinal cord injury. So that's evidence that there may be some residual descending motor connections innervating the lower body. Another way of doing that, going from the bottom up, is to do fMRI and stimulate the feet. So in about 50% of patients with ASA complete injuries, no sensation and no motor function at all in their feet, they actually get somatotypically correct, or the right area of the brain gets activated when you squeeze their toes or when you take a paintbrush to their toes. So that's evidence that there may be some connections still coming through, and maybe that's something we can potentiate and enhance. One mechanism that we think is driving this is demyelination of the cord. So when an axon is injured, or when there's reduced blood supply, for example, you start to lose a lot of the myelin sheets that's necessary to have rapid communication through the axon. And when this happens, you get exposure of these potassium channels that are typically hidden by the myelin. The myelin is removed, so the potassium channels are re-expressed. And by labeling a drug with F18, we can actually measure the expression of these exposed potassium channels. So we have something called 3F4AP, which is Ampyra, which is a multiple sclerosis drug that's been labeled with F18. So now we can track what axons are demyelinated, both in the brain and in the cord. Here is a rat study. So you can kind of see the injury model here. There's a slight laminectomy right there at T10. And this is the early and the late PET tracer uptake. So if we zoom in on the injury, we see this very high uptake of the 3A4AP tracer right at the spot of the injury. And the cool thing is that this is not the acute response. So this is not the neuroinflammatory microglial response. So at 2 days post-injury, when you would expect maybe to have the highest neuroinflammation and turnover, there's no increase, or barely any increase in demyelination. Whereas 7 days later, or up to 4 weeks later, we have this peak of demyelination that might actually be spreading across the cord to T9 and T11. So we see that this region is being demyelinated. It's still alive, so it's still viable. So maybe this is, a nice early target for interventions. We now have the tracer approved in human use. We've only tried 2 subjects thus far. The first was a young man who had a rock climbing incident, complete crush of T12. And unfortunately, this hardware that's implanted limits our ability to use MRI to see the injury itself. But on the PET here, you can kind of see that this is the region of the hardware. And looking at the PET signal, first thing you'll notice is in the very early PET frames, which is basically blood flow, that's when the tracer is washing around in the body before you actually have binding to the potassium channels, we see this marked decrease in blood flow below the level of the injury. So that might be actually a treatment target. Why isn't the cord receiving enough blood flow? But looking later in the PET image, we don't see any evidence of demyelination. So I think what's happened here is this is a very complete injury. So all the axons are actually gone already. So there's no residual demyelinated axons anymore. And incidentally, this subject does not have that toe stimulation response. So when we squeeze his toes, there's no activation in his somatosensory cortex. Our second subject had a spinal stroke, a slight bleed here, and 7.5 years post-injury, you can still see this, the consequences of this stroke. And the interesting part about this patient is he's seen almost complete recovery. He is now running short distances and walking almost without disability. So this is maybe the other extreme end of the spectrum, where the cord is almost fully recovered. And zooming in a little bit on this section right here, we do see that the cord maybe still has some demyelination. So I think this is a new way of measuring a residual function, and it could be a very important treatment target. So, can we use this type of imaging to detect demyelination as an early target in the first couple of weeks after an injury? can we perhaps stratify patients using imaging to say this is somebody who would benefit from a remyelinating therapy like Ampura, for example. And then, in the context of spinal cord stimulation, when we see these nice effects of spinal cord stim, either subdural or transcutaneous, that actually translates to other motor function recovery and some sensory function recovery, can we see that the cord is now getting remyelinated? So to measure the functional effects of that. The other approach we have been using, or our primary approach, is exercise as the one silver bullet for most conditions, actually. So we know that aerobic exercise can actually grow your hippocampus in able-bodied individuals. And we think this is neurogenesis that's been demonstrated in rodent models. We also know that we see a reduction in the microglial response after exercise. So it has an anti-inflammatory effect. First study, this is with Leslie Morse, and this was done at Craig Hospital. So 6 months of exoskeleton gait training. And we see that in terms of gray matter volume in the brain. So we can't really see much in the cord due to the hardware implants, but we see that the brain has a nice response. We see a growth of the caudate and an increase in gray matter volume of the caudate post-intervention or post-treatment. And looking at the kind of dose response effect, we see that the longer time the patients spent gating in the exoskeleton, the greater volume increases they saw in the caudate. So we think it's a kind of a just do it thing, that the more you use the device, the more of a brain growth you can induce. In Boston, we're not using exoskeleton, we're using FES rowing, which has a very nice profile of it. It's very safe and stable for the subject, but they still get a very thorough full body exercise. So you engage a lot of muscle mass, so you can really drive cardiac function and have a very complete exercise. And patients really like it as well. You see the smile on his face here. First time seeing his legs move, 6 months after an injury. We have an ongoing clinical trial where we see, benefits of FES rowing. We've had some recruitment difficulties in the study, but we do see treatment effect. So first of all, the more people row, the less depressed they are. And again, it's not a function of increased cardiovascular capacity. It's more just do it. Just come into the clinic, do it as often, do it 3 times a week. And with that, we see a reduction in depression in the subjects. In terms of the hippocampal neurogenesis, we have some evidence that this is 2 subjects that the red spots here, that's the hippocampus right there. So that seems to be getting larger after 12 weeks of exercise treatment. And at the same time, we see these massive increases in the VO2 max capacity and also pain reductions from a 4 to a 0. So not really severe pain, but it's gone with the exercise. Our second subject also sees some evidence of hippocampal increases post-intervention. He didn't have quite as good of an effect on his VO2 max capacity, and his pain went from a 3 on a daily basis to a 1, but we still see that the hippocampus seems to be getting bigger. And very preliminarily, if we look at the microglial response, so this is a tracer that binds to reactive microglia that are expressing a protein on their surface. That's where our tracer binds to. We see that the level of microglial activity or neuroinflammation in the hippocampus is also decreased by the rowing intervention. Finally, I want to talk to you about some work we're doing with Swedish Medical Center in Denver. This is Dr. Scott Falke, a neurosurgeon, also together with Leslie Morse, and we're looking at, what is the origin of below-level neuropathic pain? So if you have a very complete spinal cord injury, why would you experience below-level pain? Is this some form of centrally generated phantom pain that's that has nothing to do with your lower body? Well, we actually don't think so anymore. So the first real strong evidence for this came from Dr. Falke's lab a couple of years ago now, where he does a dorsal root entry zone ablation. And this is all done below the level of the injury. So you have patients with fully complete, like surgically verified, complete lesions of the cord. And then you go in below the level of the injury, and you ablate these hyperactive dorsal roots or dorsal root entry zones in the cord. So the way it's done is an electrode is put into the dorsal root, and if it's hyperactive, basically epileptic inactivity like this, it is ablated until the activity is removed. You move on to the next dorsal root, so it's a very invasive and time-consuming surgery. But we see excellent results in terms of neuropathic pain relief from this. So a smaller study, 8 patients with very high levels of pain, 8 to 10 on a daily basis, almost at the suicidal level, completely pharmacologically resistant. They were all on opioids, but they didn't really help. And all patients had complete thoracic level injuries, and we collected resting state fMRI pre and post injury. And I should say that the majority of the DRESS interventions was done below the level of injury. Some were done at level injury, and some slightly above the level of injury. But the absolute majority is done where there should be no innervation. Complete pain relief in 7 out of our 8 participants. The 8th one had some residual pain in his feet, but all of the rectal and genital pain was eliminated by the intervention. So very successful treatment in these 8 subjects. And also, I'm not sure if you can see on the slide here, but also the emotional aspects of pain, so like pain catastrophizing or kinesiophobia, all had significant reductions. So this is, as I'm from the psychology field, but if you remove the source of the pain, then the psychology recovers immediately. So you have reductions in depression and reductions in this fear of movement and such. So what's happening at the brain level? Well, we see this massive increase post-operatively in the functional connectivity of the primary somatosensory cortex and its projections or connectivity to medial prefrontal, hippocampal, and amygdala regions. And if we break this down a little bit more and looking at, so this is a group of healthy subjects. This is the connectivity between S1 and this medial prefrontal region. So in healthy subjects, there is some positive connectivity. In spinal cord injured subjects that are matched but are basically pain-free, there is a negative connectivity or no connectivity, whereas in the painful neuropathic pain spinal cord injured subjects, we see this negative connectivity, so this anti-correlation between those two regions preoperatively. And postoperatively, we see a massive increase in the connectivity, so they're from anti-correlated back to more of a non-correlated state. And this also implicates other brain regions. So this is the nucleus accumbens, which is a sort of a reward learning center in the connectivity, again, from the medial somatosensory cortex, which is where we think the lower body is represented. It goes from negatively connected to positively connected post-intervention. So somehow all these interventions that are done below the level of the injury have massive brain effects and also massive pain relief. So our working model is that there's actually pain transmission through the sympathetic efferents. So the pain signal, we have a hyperactive dorsal root. That signal gets routed from the lower body region across, around the lesion up to the cord, and then reaches the brain either through sympathetic efferents that reach the that travel all the way up the sympathetic system, or maybe they go back into the cord and reach the brain that way. So I think what is going on is we have this disbalance between the traditional spinothalamic tracts that is our primary pain sensation system, and then we have this parallel system of sympathetic efferents that also innervates the brain in the normal case. And if we have disruption between the two and a disagreement between those two signals coming in, the brain then interprets that as a neuropathic pain. So that's kind of a new model and maybe some new treatment targets for deep brain stimulation or spinal cord stimulation or sympathetic stimulation. So to conclude, demyelination is a nice target that hasn't really been looked at that much in spinal cord injury. The exercise interventions are really helpful, and I think we've also demonstrated that we can track these treatment-induced changes, especially in brain structure and brain volume increases with the interventions. Then we have this hypothesis about sympathetic innervation that's transmitting neuropathic pain, or the experience of neuropathic pain from below the level of lesion, reaching the brain through going around the cord. So in summary, these tools are, I think, very helpful for finding new ways to understand the extent of an injury and maybe finding new treatment targets and then monitoring how can these treatments, how can our different treatments be tracked in a more objective way rather than just self-report, for example. So with that, I'd like to thank my team of postdocs, Carl, Jesse, and Mario. So Jesse is now a faculty member at the University of Minnesota. Pedro, who is behind the 3F4AP, the Ampura, the labeled Ampura and the PET imaging there. Leslie Morse, who has been instrumental in a lot of this collaboration, especially with Scott Falke at Swedish Hospital. And Anne van der Winkel, who I didn't mention that, but we're also looking at behavioral interventions to see how can we track those with neuroimaging outcomes. And she's also here at the University of Minnesota. So thank you.
Good afternoon, everyone. I'm Ceren, Yarrar-Fisher. I'm an associate professor in the Department of Physical Medicine and Rehab and Neuroscience at The Ohio State University. And currently, I am in academia, but I may lose my job and become an influencer in TikTok because of the questions I receive about microbiome, nutrition, and metabolism, and Ozempic lately. So you may see me in TikTok very soon. That's right. So we started ProSci Lab a long time ago. We changed the name when I started my position at Ohio State. And this is going to be a different presentation from what you have seen so far. So I apologize if you're bored. But we really focus on modification of nutrition, electrical stimulation, and microbiome for improving metabolic function, neurorecovery, muscle biology, and blood pressure. So we approach SCI as a systemic disease in our laboratory, and that's what we've done for the last 10 years, I would say. My other role is directing clinical research in Belfort Center for Spinal Cord Injury. Phil Popovich and Dana McTeck started this several years ago. They're good friends, and they convinced me that this is a good idea to come and join them. Jan Schwab also joined a couple years before me. I really like the methods we do in the center because we approach spinal cord injury, as I said, as a systemic disease. And then we have parallel models of human clinical trials and animal models that we answer questions at the same time simultaneously. So that's a very impactful model, and we can talk about it later since we won't have time today. And I titled my talk as Microbiome Makes You a Superhuman or a Diseased Human, and that's very true in many perspectives. So when you think about humans are super organisms, because if an alien land right now and come to you and ask what you are not human in their perspective, you're a super organism. Because you or we evolve with microbes, we actually co-evolve with them. We have trillions of microbes live on and in us, and we rely on a lot of functions that our human genome cannot encode for. So what you see on the right side is the bacterial data on human skin, and the very right side is the molecular data. There are the molecules produced by the microbiome. What you can see is that the red is high diversity, blue is low diversity. Even in the areas you have low diversity from bacteria, you have high diversity of the molecules they produced. That's incredible. This study is very instrumental, I think, understanding also how microbiome overshadows our human genome in terms of genetic capacity. They took 1,200 samples from Europeans, Asians, and Americans, and they run shotgun metagenomics. And they found about 10 million microbial genes in 1,200 samples. And I want you to think about that. This is incredible. This is an ancillary microbial genome that we carry around us. These genes are not silent. They're active. They have lots of functions in nutrient absorption, in your immunity, in digesting your food, in drug interactions. So we need to think about microbiome as the superhuman power. Now, it makes us diseased as well. If you watch Fight Club, which is one of my favorite movies, so we're going to remember from this presentation what happens in Fight Club stays in Fight Club. What happens in gut microbiome or in your intestines doesn't stay there. So it's the opposite of motto of Fight Club. It goes into the circulation. It goes into different organ systems. It causes chronic inflammation. There are lots of accumulating evidence from animal and human models. These are very strong data around the world, not only the United States, showing that cancer is related to microbiome composition and function. Metabolic dysfunction, autoimmune diseases, inflammatory bowel diseases, they're all related to microbiome. And we are now understanding the function of microbiome better than in the past. So why did we start doing microbiome research? I want to remind you, I trained as a physical therapist years ago. I worked in neurotrauma for 1.5 years, went back, got my PhD in neuromechanics, and did my postdoc for 4 years in a big nutrition, metabolism, and muscle biology lab. So I'm like a kitchen sink for data and for multidisciplinary research. So when we were really interested in metabolic function, there were lots of studies coming from Phil Popovich's lab, and I was starting my postdoc, and he was showing that there is gut dysbiosis within a couple weeks after injury in animal models. And when you think about gut dysbiosis, that's the imbalance in the communities of microbiome. The commensal beneficial bacteria are taken over by pathogenic bacteria, and that happens very quickly. And then we actually reproduce this data in human models many times. Injury level and severity affect the magnitude of gut dysbiosis. We also reproduce data in humans. There are lots of depressive and anxiety-like behaviors related to microbiome changes after injury. And changes in bacterial taxa post-SCI predict development of intestinal inflammation and impaired capacity of nutrient harvesting within the intestine. And nutrient harvesting is the key word for us, because when it is dysregulated, the metabolic function, the neural recovery also is impaired after spinal cord injury. So that was good reason to start microbiome research, and at that time I was in Alabama. We decided that bacteria have more culture compared to local Alabamians. Hopefully there's no one here from Alabama. So how do we study microbiome? We started with cultures. If you remember, we would take bacteria from its own environment and we would feed them and they would grow. But it became very limited because we don't know every bacteria, how to feed them, what they subsist on. So we turned into molecular techniques like 16S ribosomal RNA and shotgun metagenomics. So the first 5 years of our work include a 16S, and that's a very targeted V4 region of the 16S gene is a very targeted approach. It's a good approach because it tells us who is there, how much of the bacteria are there, but it doesn't really tell us what is the function of the genes of the bacteria, right? So we were limited also in the resolution in terms of we can see up to genus level, but the species level is really not shown in 16S. So within the last 3 years, we started using shotgun metagenomics, and I think the science improved quite a bit after that in terms of the claims we can make about the microbiome impact on SCI. You can also use metatranscriptomics to understand what is transcribed off the genome of bacteria. You can use metaproteomics or metabolomics, like small omics techniques, which we use metabolomics with for the last 5 years. That it provides incredible amount of data. Okay, let's go through some studies. And we have a lot more data than this. I'm going to show you about 4 papers we published, kind of reproducing the data we have shown in animal models, but we have a lot more coming. So our first study really focused on understanding the difference between gut microbiome composition between people with acute level injury, acute injuries, long-term injuries, and sex, age, and geography match able-bodied controls. If you do microbiome studies, you have to consider people from the same geography and hopefully similar nutrition, because that changes a lot from America, North America to South America, or different countries. So we wanted to really match the geography. So the fancy graphs you're seeing is pretty much showing you how groups behave differently. It's a principal component analysis. So when you look at these colors, I'm trying to remember, red is able-bodied, black is long-term SCI, blue is acute SCI. They're clearly doing something different. They have different microbiome composition. So when we look at the results, there were significant differences in alpha and beta diversity. And when you talk about microbiome, everybody throws alpha and beta diversity first because it's an important measure. Alpha diversity tells you the richness, which means how many, the total number of bacteria, and evenness of bacteria, how they're evenly distributed. Because in an unhealthy microbiome, nothing is evenly distributed. There is pathogenic bacteria taking over, so you see mostly pathogenic bacteria. When you talk about beta diversity, it means it's the dissimilarity between populations. Alpha diversity is really telling you the one system, one organism. Beta diversity is comparing 2 different or 3 different populations with each other. So in summary, what we have seen is that SCI, both acute and long-term, have microbiome features linked to metabolic syndrome, and we define which populations are those. Inflammation-related bowel disorders, depressive disorders, of course, antibiotics use, related bacteria. Also, the long-term SCI had, as you expect, extreme physical deconditioning, and the bacteria kind of mimic that. And I always receive this question, the first thing. They use antibiotics. It could be antibiotics. We don't know. It could be. Or we can think like this. It could be antibiotics plus SCI, which is worse. So we need to think about this combinational therapies, how we can actually target not using many antibiotics and improving microbiome at the same time in the acute level of injuries. The second study we published looked at, SCI has some metabolic dysfunction, right? So what we have shown in the lab was that it doesn't matter you're 18 or 65 when you have your injury. Within 3 years, you develop insulin resistance, pretty much 80 to 90% of the time. And that insulin resistance, we thought, come from extreme muscle loss, extreme physical deconditioning, neurogenic obesity. Again, we published all these papers over the years. You can PubMed easily. But we wanted to see, is there a healthy phenotype within SCI individuals? So we took about, I think, trying to figure out the numbers. I think we took about 25 people and divided them into metabolically healthy and metabolically unhealthy phenotypes. And we wanted to see the differences between 2 phenotypes in terms of microbiome composition. And the boxes are, again, it's too small for you. The reason I boxed them out was that they were metabolically really different. They are glucose, insulin, pro-inflammatory cytokines, and some of the lipid profiles were different. That's how we phenotyped 2 groups. So, this one, the opposite. Okay. So what we have shown after this study is that there were communities if I can manage this, that are really different in individuals with metabolically unhealthy phenotype. So for example, clostridales were reduced in the metabolically unhealthy phenotype as opposed to akkermansia, which was increased in the metabolically unhealthy phenotype. So when you start thinking about different populations of microbiome, the only data we have is going back to these big studies and understanding the function of that bacteria. This is what we do. We go to big cardiovascular, obesity, diabetes studies and understand how these populations were shown in there. According to the existing data, what we understood is that the higher, the lower Clostridia in the system actually is due to diet or the changes after spinal cord. This bacteria produces short-chain fatty acid. They're incredibly important for the gut integrity. Remember the Fight Club motto. If the bacteria goes into circulation, it impacts other systems negatively. By keeping the gut integrity healthy, you actually limit bacteria translocated into different systems. But metabolically unhealthy profile or the phenotype did not have protection. So that was really important to show. Another thing was really interesting is we did untargeted metabolomics analysis. So we are going into this very unsupervised. We don't have any hypothesis saying this is changing in them because we saw in other studies. We wanted to just take a step back and see what is changing. So two important gut-derived metabolites came out from our untargeted metabolomics analysis. Again, very rich literature in cardiovascular and obesity studies. So indoxyl sulfate is the end product of microbial fermentation of tryptophan to indole, followed by endogenous oxidation and sulfate conjugation in the liver. And when you look at the literature, it promotes vascular endothelial damage via activation of inflammation. It's everywhere in obesity and cardiovascular literature. Another gut-derived metabolite we have found was phenylacetylglutamine. And phenylacetylglutamine results from liver glutamine conjugation of phenylacetic acid, which is almost exclusively derived from the microbial conversion of phenylaniline. So you get it from your diet and you convert it in your liver. And it was really high in people with unhealthy phenotypes. And that increases the risk of cardiovascular disease by enhancing platelet reactivity. Again, there's a very rich literature in cardiovascular studies showing phenylacetylglutamine is the major player. Okay, so we have done also impactful nutrition clinical trials. This study was Neidler-funded trial. And I'm not going to show you the metabolic part of this trial, but we have shown that by using low carbohydrate, high protein diet with healthy fiber amount, you can correct unhealthy metabolic state, which is insulin resistance and prediabetes, into a normal state. The data is very strong. This data was done in a pilot study and then in a bigger clinical trial. The results are almost the same. And we fed individuals 8 weeks with the diet we prepared in Metabolic Kitchen. Everything was controlled, and the control group received their standard diet, and that was followed also for 8 weeks. This is how the microbiome behaved. The microbiome is very changeable, so don't believe people say, well, you burn with a microbiome and it will never change. It's not actually true. The disease, antibiotics, spinal cord injury, traumatic brain injury, now we have lots of data showing that it changes very quickly. And it doesn't go back to the old standards very also easily. So the diet group, as you see on the left panel, and the standard diet totally behave differently. What we have shown is that the several microbial communities that are involved in community stability, decreasing intestinal inflammation, maintaining gut barrier integrity, promoting mucus production, and modulating immune responses increase in the diet, and that's incredible. And also, the bacteria that is related to metabolic disorders, cardiovascular disorders, decrease in the diet group. This was, again, very impactful finding in my opinion. But the best finding is this. When you do untargeted analysis and you're unbiased, you find something in your data set, and then you find the same thing again after doing an intervention, and that thing actually decreased with a healthy diet is a very impactful result in my opinion, and I'm very biased. But phenylacetylglutamine decreased almost very significantly, and you think about our sample sizes are really small compared to all these population studies done in other parts of the world. But phenylacetylglutamine decreased quite a bit with the diet we used, but the standard group did not change. Okay, last piece of data before I bore you too much. This is one of the things I'm very excited about, utilizing metagenomics and understanding the relationship with metabolic health and microbiome function. With metagenomics, we can actually understand who is there at the species level, but what is their role in there? So in this study, we have 46 participants, and then we wanted to understand how their metabolic profile clinically compares to their microbiome profile. We measured fasting, glucose and insulin. We gave a glucose challenge. We also measure insulin resistance, insulin sensitivity, hepatic insulin extraction, lipid profiles, pro-inflammatory profiles. So what we have shown is that microbes related to complex carbohydrate degradation and maintenance of gut integrity are implicated in the better metabolic health. So they were correlated with better insulin sensitivity, better insulin under the curve, glucose under the curve, better hepatic extraction, insulin extraction. And harmful microbial metabolites and gut inflammation are linked with worse metabolic health. So this is really important because now we're going to start talking about targets. Now we're going to start talking about developing probiotics to target certain functional pathways to improve something. We are kind of getting there. Are we there? No, we are not there. But maybe in the next 5 years, we'll be there. And this will also help the whole system. This will help neuro recovery. This will help neuroinflammation and many things we cannot think of. And upcoming data, actually they're under review and in preparation, is looking at how diet changes functional pathways. We have a clinical trial ongoing with ketogenic diet, which is another story. It's an NIH-funded trial, which we're very excited about. Again, we did the safety and feasibility trial first. That trial targets neurorecovery. Can we find neurorecovery targets in the microbiome that we can target with symbiotics, probiotics? So that's what we are after. So we're going to publish our first microbiome data from that nutrition trial within the next couple of months. And we're very excited about also starting this new work with microbiome function and bowel function. And thanks to Matthew Roderick, I don't know if he's here still, but he brought Third Frontier funding to Ohio. And we got the money now to pursue bowel function and microbiome this year, and it will be very impactful. And this time, we will do data integration of multiple omics, metagenomics to get a better picture. Okay, the big picture. We have an ancillary microbial genome that influences our health. Microbiome expands the genetic and functional capacity of the human host. What happens in the gut does not stay in the gut. Gut dysbiosis post-SCI through some mechanism adversely affects host physiology, and it changes very quickly after injury. And microbial-derived metabolites definitely influence metabolic function, and that was the result I show you. I'm not going to read it again. And a healthy diet improves microbiome, and that is for sure. Okay, so the last slide. What is waiting for us in the future? So fecal transplant therapy is a very common thing for C. difficile patients. If you think about it, if you take a healthy microbiome and transfer it to a diseased population, they can fix C. difficile 90% of the time. And antibiotics only work from 40 to 60% of the time. If you're lucky, maybe 60% of the time. So it's a very impactful way of doing things, fecal transplants. I am more excited about the bacteriophage therapy. They are bacterial viruses that kills targeted bacteria. So which means your patient have to go through broad spectrum of antibiotics, and we don't want you to delete all these good commensal bacteria that could help with neurorecovery. Now we can target certain bacteria to kill them and then keep the actual commensal ones alive. That is possible. There are actual studies already going with the bacteriophage therapy. Can we genetically modify bacteria? We can. There are labs doing it already. We can actually target certain bacteria to increase anti-inflammatory effects within the intestine so you can decrease the inflammation. Can we combine diet with microbial cocktails? That's possible. Nutrition is always the low-hanging fruit once it's used in a scientific, controlled, and accurate manner. I want to thank all my lab members. They have been instrumental doing this project. Of course, the funders who actually funded our work so we could actually move forward. All the centers that help. I was at UAB in the past before going to Ohio, and then some of the work I show you came from UAB. So we are definitely thankful for our participants. Last slide. It's kind of worthless to say lived experience, lived experience. We have been saying it for the last 10 years. Science is just catching up with it, maybe within the last 2 years. It's incredibly important. I think the conversation has to go on with the people with lived experience, but I think art should be the common conversation methodology when it comes to lived experience. I'm a sucker for independent movies. My best friend has a spinal cord injury. He made a movie. There were other friends, community members who made movies of spinal cord injury. Please, please watch them. That will tell you a lot more about the lived experience than sending a survey or getting your grant reviewed 1 week before the due date. That's it. Oh. That's my kids making kombucha because we're healthy in microbiome.
Well, hello everyone. My name is Dimitry Sayenko, and if you guys wondering about the title, I'm still wondering about it. I was just probably got too excited being invited to this session. Thank you very much. Okay, so I'm going to get back a little bit to spinal stimulation and talk a little bit about it, and maybe kind of the overall purpose is to steer some thoughts and maybe come up to certain, maybe a little bit of more understanding of what is going on in the field. So I will cover a little bit history. We'll try to synthesize them, give you some examples, and then share some recent data that we recorded in my lab. So the premise of spinal stimulation, I think, can be boiled down to the three principles. So first of all is that, you know very well, communication with the brain is interrupted after spinal cord injury. Also, we know and we saw it exploring different options that spinal circuits still remain functional below the lesion. And also, we have to keep in mind that in almost 80% of people with spinal cord injury, even if they are diagnosed with clinically complete injury, there are some spared pathways, though subfunctional. Now, the mechanism of spinal stimulation was very well demonstrated and depicted by my very good friend and colleague, Juliana Takalo, in his review 2018. And it basically premises that in the intact spinal cord, there are varieties and combinations of interneurons which work in coordination, and some can be combined in central pattern generators. There are two very required mechanisms or components to make them functional. It's descending input to initiate the certain thing and certain action, and also the sensory afferent input. After spinal cord injury, descending input cannot reach to these motor neurons or interneurons. And even though partially some activity can be activated or initiated using sensory input, this activity and output is quite subfunctional. In the presence of spinal stimulation, however, we can pre-activate, pre-excite these pools and interneurons, and then they become responsive to the descending command, and even provide more, can generate more meaningful, functionally relevant pattern during sensory stimulation. And of course, in humans clinical trials, there was lots of hopes and hypotheses and expectations that we can repair, regenerate, regain, and very little, we know how little happened. However, lots of things also happened. And there was a seminal study led by Susie Harkema, Yuri Gerasimenko, Reggie Edgerton, and other talented people that demonstrated that in person with motor complete paralysis, we can achieve, after a significant amount of training, independent standing and also promote voluntary motion. I had a privilege working in Susie's lab during a trial with the fourth participant, and you can see that the performance was quite impressive. So my take of this was that scientists can learn, how to improve certain things as well, not only spinal cord. So later on, I had access to lots of data from the epidural stimulation trials, and I was particularly interested in electrophysiological mechanisms and targeted activation of different motor pools during epidural stimulation. And one of my earlier works demonstrated that, yes, we can quite selectively stimulate the proximal and distal, motor pools project into proximal and distal leg muscles, and they quite responsive. So depending on the parameters of stimulation, we can get location, frequency, intensity, specificity. We can modulate this output generated by the leg muscles depending on the sensory environment, and also that the system and the network is quite complex. It doesn't only include stimulus response, so monosynaptic connections, but also involve oligosynaptic, polysynaptic connections. Later on, we performed a study with Mayo Clinic, and I actually show pictures only of the first authors, so I apologize. There was an incredible team of incredible people working in this trial. But here in these examples, you can see how pretty stereotyped, not very specific stimulation can promote voluntary function and also independent standing. So basically, you can see both examples of left and right leg movement with the field was pretty narrow, but then independent standing with quite a wider field. But the intensity and frequency of stimulation stayed the same. And this is a very important thing which was led and emphasized and introduced really by Reggie Edgerton yesterday, so which we can summarize as enabling effects of spinal stimulation. We also learned that after more than 40 weeks of training, the functional effects may be quite dramatic, leading to independent stepping. But also what is important here is that this stimulation pattern is also quite stereotypical. So we do not promote individual motor pools. We do not activate them. We provide the field, and then with a little variation in intensity, we can observe both stepping, standing, or no movement, all by volition, by intention of the person. Another approach is Swiss precision approach, which we listened a little bit today and witnessed, and it's very impressive, and it's based on the pattern recognition of healthy spinal cord and generation of locomotion, and it can be harnessed to provide targeted stimulation, which is very spatially and temporally coded, but definitely lead to very impressive results. This is an example with transcutaneous stimulation, and I would say this is, and by the way, so the previous was inducing effect, so let's call it inducing effect. And this is an example of transcutaneous spinal stimulation, which somewhere a hybrid approach, so we can see both enabling action and inducing action, right? So this is a person with complete motor and sensory complete paralysis, standing independently in a standing frame. We can also see enabling effects of transcutaneous stimulation. So this person has a task to also sensory motor complete paralysis, the person has been asked to perform rhythmic motions, and you can see quite impressive contractions of the of one leg at least. Now, this data can be summarized in the following schematics, and we came to these schematics, both Rebecca Martin and Megan Van Straten at one of the Asia symposiums. So when we can see that after SCI, even with prolonged time, even after intensive training, there is very little functional gain can be achieved. In the presence of spinal neuromodulation, the functional gain can be dramatic, extensive. We can see immediate grasp strength improvement or independent standing, although with the time and with the amount of training, we can also witness very dramatic results. In lots of cases, we see that the level of spinal neuromodulation provided can be decreased with time to achieve the same functions, and sometimes can be even independent from stimulation. However, there are still lots of questions, and in last year, like exactly a year ago, there was a symposium, and that was very fun experience, and which led me to some questions. Actually, was a theme of the session I participated in, which is how do we have to administer spinal stimulation? Where? How focused? And so on. So, you know, like it's kind of like a thing that I can go on and on and on. So lots of questions from this. But this led me to some preclinical model experiments where we decided to take a look in a pig study, in a large preclinical model animal study. And our team managed to have experiments, again, in Yucatan pigs, where we implanted a clinically graded array above the lumbosacral enlargement. We mapped the responses using these selective patterns of stimulation. So we knew where we are exactly. We were exactly above the lumbosacral enlargement, where there are motor pools projecting to hind limb muscles. Now, let me lead you a little bit more into the depth of neurophysiology here. So what we also did, so this is this implanted epidural array. Here, the intracranial screws that we implanted above the motor cortex of the pig. We measured electromyography muscle signals from both forelimbs and hind limb muscles. And in pigs, it's a little bit tricky. Sometimes in humans as well, using electrical current to evoke responses in both in hind limb muscles or forelimb muscles. So we have to apply not one single pulse, but a train of 5 pulses. So just remember this one. And also we first systematically investigated and recorded a recruitment curve. So this is where we applied stimulation of different intensities, gradually increasing it. And these are examples of responses in the hind limb muscles at motor threshold. You can see that there are barely responses here, right? Now, with this stimulus intensity progression, we can get quite large responses, right? But for our experiment, we titrated it down to motor threshold. Now, we applied this cortical stimulation with frequency of 1 Hertz. And at the same time, we applied very low intensity epidural stimulation of 30 Hertz. Now, when this 1 Hertz and 30 Hertz interplay, they not precisely match each other. So there is this disconnect or desynchronization between arrival of cortical pulse and epidural stimulation pulse, right? So there is a progression in time. Please stay with me, it's almost done. Now, the hypothesis was that at some sweet spot, at some time, at point in time, those impulses or pulses from the brain will collide with epidural stimulation. And based on the central conduction time calculation, we kind of, in this animal that was calculated as 17 milliseconds. So the hypothesis was when they collide, this would be the perfect time when they arrive together to the same motor pools, and then we see a larger response, right? You with me? Okay, now, So let me walk you through the results of this. So these are examples when this motor evoked potential induced by cortical stimulation, and the red arrow indicates when epidural pulse arrives, right? And you can see that the closer they are to this magic sweet spot, the larger responses get in, right? And the intensity of stimulation is the same. It's motor threshold or even submotor threshold, but you can see the huge facilitation of these responses when descending pulses arrive at the same time with epidural stimulation. It's beautiful, so you can see 10, 100 times more facilitation that compared to the motor threshold. And what is the, let's put it, a sad part, bad news, is that once the epidural stimulation stopped, these motor evoked responses, motor evoked potentials stay pretty much the same. They come back to the same level, right? So speaking about the carryover effects, well, it was a very quick experiment. It was several minutes, but unfortunately, we didn't see much of the carryover effects or potentiation or long-term, short-term plasticity or potentiation. But what we saw when we modulating the epidural stimulation intensity, so we used this motor threshold descending, motor threshold cortical stimulation to sort of mimic an incomplete spinal cord injury, right? Because when the stimulus, the full strength stimulus arrives to the spinal cord, but like a little bit shrinking down. So this is our motor threshold cortical stimulation responses represent. But then we titrated, we used a little bit lower or higher epidural stimulation pulses. And you can see there is a huge dependency on the intensity of epidural stimulation, with the peak of facilitation seen about at one motor threshold, so equal to one motor threshold. So the closer we are to motor threshold using spinal stimulation, the better gain. And you can see the gain can be quite significant. Now, wrapping up, we knew that functional improvement can happen during spinal stimulation. But I would like to draw your attention and maybe look together at this as enabling and inducing, like an enabling or inducing approach. But I would also like to emphasize that according to what we saw, the extent of physiological and functional effects really depends on the intensity of spinal stimulation. And it leads me to the suggestion that we'll talk a lot about quantification and standardization of clinical outcomes after longitudinal clinical trials. But I would really look at this as also quantify and standardize the stimulation parameters. At least we can relate it to the motor thresholds. And of course, there are questions, right? So we were, I think, focused a long time on the spinal automaticity and looking into the mechanisms, electrophysiological mechanisms within the spinal cord. I thought myself, and this goes through all my study, these monosynaptic pathways when I stimulate spinal cord and see responses in different muscles, kind of like under premise that they could explain us everything. But in fact, I think what is really important is the whole system, and that's why in this session, is the interaction between not only brain, but descending commands with spinal motor pulse, which is extremely hard to investigate in humans. So we have to recognize it. And then just a question, maybe the discrepancy between functional effects, again, in clinical trials that we saw so far, can be attributed to the intensity of stimulation at least, right? And if you think about it, if we read the paper and see that there was a population of 100 people participating in the study, stimulation intensity was between 5 milliamp to 200 milliamp. We cannot expect, there is no way we can expect the same mechanisms and the same effects in this population. So we have to at least probably group them in relation to motor threshold, at least. So this is it. This is my beautiful team, my collaborators, the grants Also, I would like to mention that we enroll in participants, so if you know someone who are interested in participating in our trial, please contact me. That's all.
Hi, everyone. My name is Yvonne Deere, and I am the person with lived experience on this panel. I did not prepare any slides, but I was really excited to be invited because, as we all know, spinal cord injury is a whole body syndrome, and we got to learn about a lot of that just now. I'm just going to ask a couple of questions to kind of get us started from these three, and I'm going to relate it to my own personal experience because somehow, luckily, that is possible. I have a T11 injury from almost 11 years ago, Asia A. I've done my own personal research, I guess, experimentation, just trying to access anything and everything that I deemed safe and reasonable over the past decade. So yeah, first, Clas. I personally have a concept rower at home, and I love to use it every week. I aim for 30 minutes a week, and I'm really proud of myself if I do an hour. You were talking, though, about pain and potentially stimulating the dorsal roots to reduce pain. Are you currently actually doing any clinical studies with that? And if so, are you talking like transcutaneous or epidural? Like, what sort of stimulation are you imagining for this?
Yeah, no, so the work I presented was actually lesioning the dorsal root entry zone. So that's something that Scott Felke is doing in his clinical practice. So we haven't, done any stimulation or, potentially like dampening this hyperactivity, but I think it's a very
dinner and bedtime for me.
Oh, yeah. So there's a phantom here somewhere.
Yeah.
I think it's a very potent region to target, so that's certainly something that I hope the neuromodulation field will look into.
Yeah, definitely. It seems like transcutaneous would be an excellent non-invasive way to explore that idea, which I'm sure a lot of people would be very happy about. And then about, I guess, 3 years into my injury, I focused on, I did acupuncture and traditional Chinese medicine to specifically try to treat like bowel and bladder, but I was at a gym where the owner worked with a pair of acupuncturists, and she was an incomplete and regained the ability to walk independently somewhat. And so I invested a whole lot of money to try it myself. It did not work for me, but it was still worth the try because, worst thing, it was some acupuncture and some really smelly mud-like water that I got to drink every day. But from that, I definitely learned a lot about different cultural aspects of what are healthy foods. I was born and raised in Texas. I think of health foods as salads and cut up fruit and raw foods a lot of the times. And from the traditional Chinese medicine perspective, that is not necessarily the case. And so for me, I've learned like cooked foods are a lot different. And even it's interesting, you showed your kids making kombucha. Kombucha goes right through me. It is not like healthy for me. Mm-hmm. Interestingly enough, red wine, also fermented, totally fine. So that's good. But I am curious if you know of any like current probiotics that you would recommend for like just a generic something for somebody with a spinal cord injury to give a shot.
Yeah, there's nothing I can suggest. And I'm very careful about when I talk about nutrition, because we use nutrition as a targeted therapy. So if you want to correct metabolic function, we want to make sure the carbohydrate, protein, and fat amount is controlled. And nutrition is very cultural for people. So I think healthy foods are commonly vegetables and less meat and less fat. That's a Mediterranean diet style that has the strongest data in the world for being healthy. But regarding probiotics, everything is very untargeted and very mainstream. Some of my friends said some of the probiotics over the counter work for them. Some says it doesn't work, so I really cannot tell you what works. The only way we can know this is, and I was going to actually talk about it but didn't have time, one of the things that we are imagining right now is to transfer spinal cord injury human microbiome into an animal and then compare it to germ-free mice and actually kind of modulate the life of the mice either with nutrition, probiotics, symbiotics, or targeted strains. So that's our goal, to understand what will work in animals first and then translate into humans. Because the way we do the science right now is really, really weak in terms of we are working around correlations or previous data collected from able-bodied individuals. That science can only be improved by utilizing both human and animal model together, which we are on it at the moment. So, but back to your question, no probiotics. There's nothing.
That's fair. And really interesting that you mentioned trying to transplant human microbiome into animals, because that was something I was thinking about during your talk about if it's, you know, geographically different. Of course, it's also different between species.
Of course. Yeah.
All right, and last, I actually just got an epidural stimulator implanted on Friday. And so I'm at the very beginning of my journey of figuring out what parameters work for me, and I am completely with you that we do not know. Like, we can't just be like, all right, here's your stand-up parameter, here's your, like, go to the bathroom parameter. And so it is interesting, though, because it's such an individual process, even where we put the stimulator, as you know. For me, since my injury is at T11, my cord is incredibly atrophied right at T11, T12, which is a common implant location. So mine's at L2, L3. And so from the research perspective, it does seem like grouping into categories of strengths of stimulation would be like a good way to think about things. But also, what do you think about the location? Should we be standardizing that for human studies, or should we maybe allow a little bit more wiggle room?
I would say that some wiggle room may be healthy, but it's pretty standardized in terms of like a very general standard of operation, right? So it's cervical enlargement for upper limbs trials and lumbar enlargement for lower limb trials. And the former is, so it's a span between C4 to T1, and the largest, the lower one is the T12 about, right? So in your case, it was lower. So I think, you know, I mentioned this before. If I have all resources in my possession, I would probably go through the inducing stimulation route, and yes, I would try to investigate the precision delivery, but I see also that wiggle room and some wider field also works. It can work. I think that if we allow to adjust right stimulation intensity at least, we can kind of compensate this precision and intensity. We can compromise. We can find a balance.
Makes sense. Cool. And that's all I have if we want to open it up to the floor.
Okay.
All right. Yeah, so, you know, this question of demyelination after spinal cord injury has been dogging the field for decades. And I was very happy to see that you have a PET ligand that could potentially be used. Looks like the proof of concept is a bit difficult in the thoracic injuries with instrumentation. So I was going to suggest that you might look at cervical spondylotic myelopathy, where we know that there's also a lot of spinal cord demyelination, or at more moderate cervical spinal cord injuries, such as what we call central cord injuries. And the reason for that is that those may not have any spinal instrumentation, and you'll get a big reduction in the artifact from movement if you focus on sort of the upper cervical spinal cord.
So I totally agree with you. I think so. We have proof of concept in MS in humans, so we know that the tracer works. There are some other complications in translating the animal studies to the humans. So one big thing is actually that the animals are under anesthesia, and the metabolism of the tracer is highly impacted by the anesthetic. So we get much nicer pictures in the animals than we can get in awake humans thus far. But certainly that's something we're trying to find that right patient population, and I agree with that.
Thank you. So that was really interesting about your pig model. And it was kind of funny that you didn't see potentiation after you stopped the cranial stimulation. I'm wondering if you attributed that to the fact that you had to use a sequence of pulses in order to evoke that response.
No, it's definitely. I think what is the very buzzword in our field is plasticity, right? And to what extent we can expect plasticity after 7 minutes of a trial, right? So I think short-term potentiation, long-term potentiation still worth to be looked into in a deeper manner. I would say so far, I would take it with a grain of salt. So I think that everything is possible, and maybe chronic experiments would be more helpful.
So I have a quick question, and then I'll pass the mic on. Very nice presentation to you all. Thank you. Ceren, I have 2 questions. The first is, how are you defining insulin resistance? Because we just finished a study funded by the Nielsen Foundation where we looked at a cross-sectional study, and our primary outcome was insulin resistance, and we're not seeing 90% incidence or prevalence, I should say, in our group. Mm-hmm. That's the first question. And then my second question is, when you describe the time frame of the onset of gut changes, it made me think of the change in osteoclastic bone resorption. Within a week after injury, there's this uncoupling of remodeling. And one question has always been disuse versus neurological injury. And I wonder if there's any literature in the disuse or the bed rest human models that you've looked at.
Mm-hmm. So the answer to the first question is we use oral glucose tolerance challenge and then use the Matsuda index, pretty much put the numbers in an algorithm, and then that will give you a number. And normally if it's lower than, I think, 2.5, there's a standard for diabetes, and then you consider them either pre-diabetic or diabetic, so there are scales. And we also use HOMA-IR for the insulin resistance. So Matsuda is insulin sensitivity, HOMA-IR for insulin resistance. One thing I want to tell you is that, so the populations we normally did studies within the first 4 years were not very diverse. There were no racial differences. We actually work on a lot of white-collar type of patients for insulin resistance. So our data is completely flipped when we do nutritional studies where we included people from all walks of life, different races. So I think sometimes we make the mistake of utilizing same patients most of the time, but the metabolic and neuro recovery profiles are so different when you go into Black individuals, Hispanics, and Asians. So our the reason I brought this up, when you look at the studies and kind of, it depends what type of races and ages and where they come from, the patient. So I think, I still believe that insulin resistance is one of the biggest threats after injury in metabolism.
Are you saying that your sample was less diverse?
In the beginning, but then our insulin resistance prevalence increased a lot when we actually have everybody in our studies from different rural areas, urban areas. And there is a big point, I think, in SCI research to be inclusive of everybody. Second question, there is a literature about extreme physical deconditioning, bed rest, and some of the space travel. Actually, we have a very big interest comparing spinal cord injury model with the space model. So bacteria changes quite a bit with the bed rest and space model. So it's not necessarily on the spinal cord injury. I think there are so many things in the system happens early on, antibiotics, deconditioning, muscle loss, and also there is a system, we believe, the hypothesis is that for why the microbiome changes is really losing that autonomic input into the enteric nervous system first, and also the visceral afferents carry feedback from the gut synapses with the interneurons in the spinal cord eventually goes to the brain. So that system is disrupted. That increases or decreases gut motility and decreases the gut integrity. There is lots of data coming from Texas actually showing the integrity is totally lost right after spinal cord injury through the enteric nervous system. But it's not the only thing. There's so many things we cannot really piece out. I don't know if it answered your question, but yeah.
Great presentations. This question is to Clas, actually. I was excited to see the increase in hippocampal gray matter volume associated with exoskeleton walking. And I believe that those individuals were motor complete. And so I'm curious as to what you think that mechanism is and where you see clinical translation, hopefully down the line.
So I will just clarify that the participants in the gating study were not motor complete. We had non-ambulatory A, B, and C. And so with that, please.
Yeah, another clarification. We didn't see increase in hippocampal volume in the exoskeleton gating. We saw that in the FES rowing. So the exoskeleton gating is not as aerobic, so maybe it's more of a motor learning function that's driving this increase in the caudate. That's, but that's neuroimaging, hand waving. So we don't have, evidence for that. But I think maybe, learning to use the device might be the driving force for the caudate increase.
Great, thanks.
Very quickly, Ceren, I just want to say thank you for your invocation of art. And very briefly, just as a proud parent of a kid with a spinal cord injury who has what I think is called a prophetic imagination due to what Bob Wagner called yesterday a savage gift, it brings it full circle to what we heard from Cynthia Kubu this morning about justice. And that art is critical for all of us to understand the story of another. And so I would doubly emphasize your point and the folks that you put up there. And then one plug that one of them, we're trying to organize a film screening in Minneapolis in June for Quinn Brett, who's one of our employees. And I would encourage you to watch for that. If we can execute it, you should come see it for that reason.
Let's thank our speakers one last time.